CDG. Okay. Please help me to welcome our returning speaker, Craig Beatty from CDG.
Well, thank you. Let's see how technologically challenged I am here. I'd like to tell you in this return visit about our platform technology, which is all based on cell-penetrating peptides. And in this particular situation, they're used as chemotherapeutic agents. Let's see. How are we doing here? Okay. Backwards? Oh. Cool. Hey, I told you I was technologically challenged. CDG Therapeutics is a small privately held company. We were founded by University of Illinois College of Medicine faculty, hence the CDG acronym for Chakravarty and Dasgupta, the same Chakravarty that's in Diamond et al. We are based in Chicago at the university. Our facilities are within the University of Illinois COM through a sponsored research agreement, and CDG Therapeutics is the sole licensee of all the technology I'm about to tell you about. We are a platform technology. This is our current research platform. We're focused on cell-penetrating peptides. We believe they're a unique platform for any number of reasons. I'd like to tell you about some. They preferentially enter cancer cells, and I will show you some of that data. They selectively rescue major pathways in cell cycle progression. They inhibit receptor kinases, in this particular case RTKs. They block ephrin receptors, both of which, the receptor kinases and the ephrin receptors, are critical to neoangiogenesis, capillary formation in a tumor. And we've recently come upon some exciting new data where our peptides interact with the tumor environment. And I'll show you examples of all of that. So, preferential entry into cancer cells. What is it we're talking about? Our lead compound is P28. 28 amino acids of the cuprodoxin azurin. Azurin itself is cell-penetrating. It is 128 amino acids long. P28 is a fragment of that. It is the protein transport domain, or PTD, of this azurin. It preferentially enters cancer cells relative to normal cells of the same type. It enters all cells, but the kinetics and the mass effect are much different in cancer cells, and I'm about to show that to you. All of this has been published. The mechanism of penetration into cancer and normal cells is both endocytotic and non-endocytotic, and we're currently exploring at some length using solid-state NMR on the non-endocytotic penetration pathway. Okay. This is simply a histogram to show you a broad range of cancer cell lines, all in vitro. From the left, melanoma. We progress through glioblastoma, CNS-type tumors, sarcomas, mesenchymal-derived tumors, breast cancers, et cetera, et cetera. The red line is unity. That would be the penetration and a normal counterpart to every one of these cell lines based on histogenetic subtype. The preferential entry overall is greater than 95%. There's at least a twofold increase over normal cells and about 75% of the cells of the same histologic subtype. I'd like to highlight initially this particular subset because they're all CNS tumors, and you'll see why in a moment. As they are able to penetrate cancer cells, and they also affect developing capillaries and small blood vessels. They don't touch larger blood vessels or the cells that comprise larger vessels. We've found them to be excellent carriers for at least contrast, and more recently for imaging agent through PET-CT. And this has been funded through recent grants through the SBIR and the STTR system from the NIH. You just, on the previous speaker, showed his pictures of what's going on with the mouse glioblastoma model. This is ours. This is simply seen from the outside. One can see the focus imagery using fluorescence, grayscale, heat maps, whatever you choose. Left to right, these are three sections of one brain, coronally through the brain, the front part of the brain, the middle part of the brain, and the hind brain. and obviously there's a tumor there that's identified by a fluorescently labeled compound. The take-home message from this is a significant time after injection of our labeled peptide. This just happens to be 24 hours, but one can go out to 48 or 72 hours and it's still there. Just to show you that it's not limited to cells that you poke into a mouse's brain, this is MRI, contrast MRI, we're using gadolinium conjugated to our P28 peptide against gadolinium alone. These are coronal sections of a pig brain. We have a melanoma model in Sinclair swine. It is hereditary, it is congenital, aka spontaneous, it does metastasize. This is what it looks like when it metastasizes to the brain. On the left, pre and post contrast using gadolinium alone. On the right, that's labeled with our peptide. Same animal, allowed for washout, put in our peptide labeled with gadolinium, pre and post contrast. The interesting difference is there are two. One, I think there's a distinct improvement in imagery with our labeled compound because it is taken up by these cancer cells, even in a pig. And these are pig cancer cells, not human cancer cells. So you're cross species here. The second thing is, and probably most important if you have renal problems and an associated a cancer, is that this, and I wish I had a pointer for this. Maybe I do. Let's see. Yeah. That's 1/80th of the dose of gadolinium over here. 1/80th on a mass basis administered IV to the animal. So, moving right along, same slide, I'd like to highlight this particular subset of cancer cell lines. These are breast cancer cell lines, and notice that one is labeled in red. This is a triple negative breast cancer. It is generated through basal cells. It's a particular type of breast cancer, histogenetic subtype of breast cancer, and it's important for a couple of reasons. One, it's very difficult to treat triple-negative breast cancer, but there seems to be a distinct relationship between this particular cancer type and the microenvironment and how the microenvironment interacts with things like chemokines. Chemokines, of course, these are a family of small cytokines or proteins, et cetera, induce directed migration in nearby responsive cells. They are part of the G protein-coupled receptor family. The interesting thing about this is recent publications would suggest that chemokines regulate basal breast cancer metastases in via the microenvironment. The chief suspect is something called CCL5, or RANTES, which is a chemokine. It is secreted by breast cancer cells themselves, correlates with progression, et cetera. CCL5 binds to its cell surface receptor, CCR5, but is also a co-receptor for HIV-1. Nothing new there. But if one begins to look at our peptide, and this is our peptide here in red, down here, one sees where it binds to the GP120 protein, which is essential for the entry of HIV virus into a T cell. This is the CD4 binding site on a T cell on GP120. Here's where CCR5, the co-receptor binding site, is. Here's where we're binding. If one continues to look at this, we find that it binds actually to the hinge region, which is distinctly associated with the co-receptor binding site here. Same model, just a different picture. And we proved this experimentally using a series of antibodies to see which one would block out the effect of our particular peptide or peptides, and found out, yes, it is binding to the V3 region. Well, that's very nice. What's that have to do with CCL5 and its receptor CCR5? This is simply on the left showing you the natural, my thumbs have to get smaller. The natural ligand here, the critical amino acid, this Phe13, binding to the blue, which is the CCR5 receptor. If we take this and take a look at where our peptide, P28, is binding, here's the critical amino acid. This is CCR5 again. You can see that it's binding to the same site. Okay, So if one continues to look across this plethora of cell lines that we looked at, and this is about half of them here on this histogram, what happens after it gets in the cell? Does it do anything? Well, it turns out it does. It does a number of things. And if we'll reflect back on the first slide I gave you, RTKs, in this case, P53, and this has been a central focus for our investigations over the last 5 years. This is simply a docking model of how P28 binds to P53, which domains of P53. Let me cut to the chase. It's the DNA binding domain. We know where it binds. We know how it binds. We know the kinetics of its binding. We know, as you'll see in a moment, the amino acids of its binding, et cetera, et cetera, et cetera. So P28 is a first-in-class peptide inhibitor of COP1 binding to P53. P28 produces a post-translational increase in wild type, and here's the take-home, mutant P53. Now, I don't have time to go into the in-depth into the mechanism, so there'll be a poster tomorrow that will show you how this compound works and how it is either affected or not affected by mutations in the P53 molecule. P28 works by binding to non-mutable motifs within the DNA binding domain of P53. It blocks this E3 ligase, COP1, to P53, which leads to an autoreduction in COP1 levels or COP1 levels, and an increase not only in the absolute content of intracellular P53, but the activity of it as well. In other words, more P53 binds to DNA, more good things happen. Okay, so this significant and sustained increase in the level and activity in P53 turns on a whole series of events downstream. Take-home message here is cyclin inhibitors P21 and P27 are activated, BOXM1 is decreased, G2M clank, the whole cell cycle stops, cancer proliferation stops. So it's a cell cycle specific agent. Of course, as more cells go through the G2M transition, more fall out and go through apoptosis. But I also mentioned that it was effective within the vascular system. RTKs and F receptors and their ligands, the ephrins. And we published on both of these. If one goes through a series of molecular modeling steps and looks at the similarity, in at least the physical similarities in this case, structural similarities, if you will, in the binding of these three, P28 to these three receptors, if you will, three proteins, P53, the vascular endothelial growth factor receptor, and the fibroblast growth factor receptor, both critical for the genesis of new capillaries in a tumor or anywhere for that matter. One sees that they bind all to what seems to be relatively the same type of pocket. In the case of VEGF-R2 and FGFR1, they are non-competitive inhibitors of the kinase activity of these particular receptors, clank at very high affinity. So if one takes this to the next step to ephrins, on the left is where ephrin A1 binds to its receptor, the FA2 receptor, on the bottom the FB2 receptor, and its ligand. On the right, in the red, this is P28, same place. Heck, how'd that happen? If one begins to look at the similarities between the various binding sites and the amino acids within P28 itself, one finds a lot of similarity in terms of hydrophobicity. Very high hydrophobic amino acid content, whatever those amino acids are, in these binding sites across these various proteins. If one looks below this at conservation of amino acids binding to proteins, these same proteins, it is absolutely astounding the conservation in this particular structure. So what's this all mean? Is it of any use to anything? Well, it turns out that it is. We've already completed a phase 1 clinical trial that we set up to do simply look at the pKa, ADMET, et cetera, and a very, very positive thing happened. Patients started to live longer. These were all stage 3 on their way to hospice patients when we started presenting our agent to them. There were no significant adverse effects. We never saw a NOEL. The MTD was never reached. It was so far above any therapeutic range that we gave, over 5 dosage ranges, there was no MTD. There was efficacy. 1 complete response, 2 partial responses, a significant amount of stable disease, long-term survival. Interestingly enough, response and overall survival was independent of P53 status, although response and overall survival were significantly correlated. In other words, if you got a response, you lived longer. And the time that you lived was significant. 3 of our patients are still alive, 2.5 years after they first entered the trial. So we're now entering a phase 1A/B clinical trial for pediatric CNS tumors, obviously. So what's the takeaway from all of this? That our therapeutic platform incorporates a series of proteins that yield peptides with similar amphipathicity, alpha helical and beta sheet domains, and low overall PI that form the basis for preferential entry into cancer and endothelial cells. The structural and conformational relationships that form these selective hydrophobic interactions, we believe, provide the basis for the therapeutic activity of peptides in our pipeline and the template for the analogs and mimetics that we've either constructed or would like to construct. Thank you for your attention.
Questions for Craig, please? Eric.
Just a further comment on the correlation between low PI and cancer cell uptake.
I'm sorry, I didn't hear the last part.
What is the correlation between low PI and cellular uptake, cancer cellular uptake? Cellular uptake, cancer
Right.
cellular uptake.
Here, let me do this. Let me try and do this. Oops. Oh, well. I was going to go back to that slide. The heck with it. The uniform characteristics that I identified in those slides are the properties of the platform. We thought we had a one-hit wonder until we began to look and how we could modify and whether we modified the amphipathicity, in other words, change the hydrophilic to neutral to hydrophobic amino acid ratio, change the chirality. We looked at everything. Those 3 or 4 properties seem to be conserved across the board, not only within this peptide, but members of this particular family that have similar types of activity. So if you're asking me, this is the drug? I don't think so. I think there's a lot out there. Fortunately, we've got patents on it. The patent portfolio is quite extensive. A low PI rather than a basic PI. The sine qua non for cell-penetrating peptides is the cationic. Rubbish. The PI on this is 4.3.
So all your CPPs are negatively charged?
Yes. The ones that we're going to develop, yes. Any others? Thanks.
Okay, please join me in thanking all of this morning's speakers.